Welcome to the Newton series. I'm Robert Wicks. I'm the VP of philanthropy at Gladstone, and we are trying this as an experiment. We've sort of sketched out 4 of these sessions with a chance for you to get to know some of our faculty, to hear about the work that we're doing beyond COVID, and just really kind of get some insight into the scientific mind and the research process. The idea for this came from Sarah Dale, who oversees the President's Council program at Gladstone. And when she pitched it to us, it was, maybe some of you saw that there was a story in the Washington Post that mentioned that Newton, while in quarantine during 1665, 1666, just underwent just a staggering period of intellectual output. And so we thought that he was a perfect patron saint for the moment that we're in. I know all of us know about Newton, but just to remind you some things about him, he was really a citizen scientist. He was born on Christmas Day in 1646, I think, 1645, died in 1727. So he really was the leading figure, or a leading figure, in ushering in this age of science, empirical science. He was born at a time when there was more belief in magic than in medicine, and more faith in witches than in doctors. And by the time he was done, I think that he basically created the reflecting microscope, invented calculus, and developed the theory of gravity. He did all of that initially, actually, just in quarantine. So from one great scientist to the next, Benoit Bruneau has offered to let us into his living room and talk about his work. Benoit, I could introduce you. Maybe better for you to introduce yourself and tell us about your academic path and how you wound up at Gladstone. Pardon me.
Oh, sure. So I am the director of the Gladstone Institute of Cardiovascular Disease. I started that new position for me on Canada Day of last year. And so I'm a proud Canadian, so it was very fortuitous to start that.
When is Canada Day?
It is July 1st.
July 1st, okay.
And so here in the US, the fireworks are always 3 days late, unfortunately. I'm Canadian, but I was born in Israel. My parents were diplomats, and so we lived all over the world for a while, and then eventually landed back in Ottawa in Canada, where I did my university, did my PhD there at the University of Ottawa, and then did a postdoc at Harvard Medical School for a few years. Landed my first job, independent position, at Sick Kids Hospital in Toronto in 2001. And then was recruited to Gladstone by our current president, then director of cardiovascular disease, defects. So I've been here for 13 years, and I've been loving it ever since.
Your topic is how genes sculpt the heart. Tell us about that and why it's important to understand that process.
So the way in the sense that I think about understanding the developing heart and understanding how our genes sculpt the heart is thinking about a blueprint, right? If you have a blueprint, you can fix something that's broken, and also with the right parts list, you can build something anew, right? So if you understand all the genetic networks that control the formation of the heart, you might understand what goes wrong when one of those genes is mutated, and what might be happening sort of downstream of that. But it's more than just a blueprint and a sum of the parts, because this blueprint, as our hearts form or as disease progresses, it morphs over time, and it sends out more and more complex instructions that feed into a complex network. Some of the instructions cancel one another out, and another one takes over. So the way that I think about it is like Rodin or Giacometti starting with a block of stone and a few simple tools, and then knowing their knowledge of whatever it is that they want to sculpt, their anatomy, their imagination. And at the end, you have a beautiful, perfect sculpture that tells you something, except that every time they do it, it comes out exactly the same shape, exactly the same size. And that only once in a while, it comes out not quite right. And so that's what I mean by how our genes sculpt the heart. Maybe I can show you a couple of examples of some of the stuff that we're doing in the lab. Yeah. So let me show you a couple of things that we've done. Let me see. Can you guys see this on?
I can see.
You guys see
your
this on
laptop? Yep.
Okay. So what am I showing you here? I'm showing you an experiment that we've done a few months ago. It started a few years ago, but this particular image on the right was captured by a cardiology fellow, a couple of clinicians in the lab, Irfan Katiria and Martin Dominguez. And what you have on the left is you guess, I don't know if you can see my cursor, but on the left you got a very early mouse embryo. This is an embryo that has exited a stage of embryogenesis known as gastrulation, when all the three germ layers come together. It's a very, very simple structure. It's the bottom part is the embryo proper, and the top part is what's going to be the extraembryonic membranes. And so this is day 7 in embryogenesis in the mouse. This is equivalent to 2 weeks in gestation in a human. So this happens very, very quickly.
Wow.
And what you can hopefully see is a whiff of purple color right where my cursor is, right? And so this purple color shows the turning on, the labeling of a gene that we've been studying in the lab that causes congenital heart disease. And we use some genetic tricks to then follow those cells and say, when you now have a formed heart, where do those cells end up? And that's what the images on the right are. And so these are microscopy images of mouse hearts at embryonic day 14, which is equivalent, I think it's probably hidden by the windows, but equivalent to human day 50, right? So not even 2 months in gestation. And these two green floppy things at the top are the atria. This big green part down here is the left ventricle, and this blue part here is the right ventricle. And there's two things to note here, is that when we've labeled these cells very early on, they go on to make very specific parts of the heart. So you can see the green cells are labeling the atria and the left ventricle, but not the right ventricle. And then these red cells are lining up right at the junction between the left and the right ventricle. When you look at the image on the back, where it says the back of the heart, you can see these red cells that are lining up super straight, like a Roman phalanx. And in experiments that we've done subsequently in a mouse model of congenital heart disease, we found that now these red cells don't align anymore. They don't line up. And so for the first time, we now have a handle on what are the cells that we need to look at to understand congenital heart disease in this particular model. And when I talked about the black box between a mutation and a heart defect, this is sort of the first glimpse, this is the first opening into that black box that we get to have. And so we're really excited about looking at this. And so what I mean by the genes that sculpt the heart is that you've gone from this tiny little blob of purple in this very simple embryo, and over the development of the heart, these cells, the green and the red cells, have been assembled into a very, very specific, very finely regulated assembly.
I mean, it's amazing that it ever works well. Why does it? What happens when it doesn't work well?
Well, when it doesn't, for example, these red cells, as we just found out, don't align properly, and that's what causes ventricular septal defects, similar to the ones that my daughter has or had.
Are those just genetic factors? I mean, what are the factors that?
Yeah, so they're genetic factors that function in what we call a gene regulatory network, genes that turn one another on. And then downstream of that, there are genes that make proteins that, for example, will help cells stick together, or will tell cells to migrate in a particular path. And when they reach their destination, something else tells them to stop. And that's how morphogenesis of all organs take place, whether it's your hands, your face, your brain, your heart. It's similar types of cues that takes place, and they're all regulated by a number of genes. And what we've learned from the genetics of human congenital heart disease is that it's a lot of the sort of the master regulators that send out the strongest instructions that are defective. So we've got this thing of sculpting But what we want to do also is we want to go live. We want to go beyond, let's see, how do I, here we go. So what I'm going to show you here is on the left is a front view, on the right is a right view of a very early mouse embryo where some cells, the broad set of cells are labeled in red, and the precursors, the cells that are going to form the heart, are labeled in green. And I'm running this movie, and you can see now that cells move. We're watching a live mouse embryo under what's called a light sheet microscope. And you can see the movement. Look at these cells kind of cranking around. There's shapes that are taking place.
What's the time lapse on this? I mean, what's the period that this is?
This is about 24 hours of development.
Wow.
And so that's at this early stage. And then if we could look at a later stage, and you can watch the early heart tube forming. So again, two different views of the same embryo. And I'm going to run this, and you're going to be able to see here this very quickly a structure forming that looks like two kind of balls, and that's the early heart tube. And so this is work that this particular movie that I just showed you, Martin Dominguez in my lab sent to me literally last week. This is absolutely brand new, and it's allowing us to look at the forming heart in a way that we've never been able to do before, because now we can label specific cells. So the green here is one particular gene that we've labeled that labels the cardiac precursors, and we're able to watch it live. And so what we're hoping to do is to understand how the genes that sculpt the heart do it, not just in looking at starting point A to point B, but looking at all the steps in between and following the cells and understanding their movement. And so we've just started doing this. And these are some of the things that I've sort of in my quarantine room, I've been, staring at these movies, staring at these data sets and thinking, gosh, what does this mean? And what can we learn from it?
You must be itching to get back and figure that out.
Absolutely.
Yeah. What is the implication of this for the rest of our biology? Whatever insights you derive are applicable to other organs, or?
Yeah, absolutely. I mean, the rules that apply to one particular organ will generally apply to other organs in the body. So the cues that made those cells move in a particular direction, or made them sort of hover around before they joined the rest of the heart, or the timing for when they would turn on a cardiac gene, those rules, which we don't fully understand at the moment, are going to be similar from one organ to the next. So, and a lot of the genes that we study are also play roles in other organs as well, right? So some of the genes that we study in the heart may have roles in forming the gut or in forming the brain, and it's sort of their network, their neighborhood that they're in that dictates sort of what they do. So for example, if you have a particular an engineer who one day is working on something over at Boeing, he's working on making a plane, and then the next day he goes down the valley and is working on a computer system. The same engineer using the same tools, the same ideas, but he's working on two different things. And so it's the same sort of thing, but we need to understand how that engineer works, what are the tools that he's working with, and how does he or she deploy them in different ways depending on what problem they're working on.
I want to save time for, I know this is just a toe in the water of what you're doing. If people are curious about it, I know that we are trying to tell this story of your research on our website and elsewhere. I think we'll have other sessions like this where we can do a deeper dive with people who are interested. Charlotte was here with her hand raised because she had really wanted to ask this question about pluripotent stem cells. Yesterday we were in the car and she said, is it possible to turn a heart cell into a pluripotent stem cell and then turn that into a brain cell? So the fact that a 9-year-old is aware of pluripotent stem cells, I think is a sign that she's been listening in on a lot of my calls. But can you say something before we go to Q&A about the impact of Shinya Yamanaka's discovery on your work in the field of science?
Absolutely. I mean, it's had an enormous impact for a number of reasons. The one is a very practical reason. So for example, one of the manuscripts that we've been working on while not being able to go to the lab is one on using the iPS cells to create, via CRISPR-Cas editing, a human model of congenital heart disease. So we're not creating an actual heart with a heart defect in it, but we're creating a cellular model where we can interrogate the gene regulatory networks that go wrong in congenital heart disease. And so without the human iPS cells, we would have never been able to do that. The other impact, though, that Shinya's discovery has had has to do with the notion of our cells being fixed in their destination. In other words, a heart cell is always going to be a heart cell no matter what. And Charlotte's absolutely right. You could take a heart cell or a skin cell or what have you and turn it into a pluripotent stem cell, or turn it directly into a brain cell. You can take a skin cell and turn it directly into a heart cell or into a brain cell. And with only a very limited set of instructions. So when I talk about the blueprint, right, what we've learned over the years collectively in understanding that the gene regulatory programs, the instructions that nature uses to make heart cells, those are the ones that Shinya induced pluripotency, and then our folks at Gladstone in my lab and Deepak's lab and elsewhere in making new heart cells for cardiac regeneration. We've been able to understand that even with a limited set of instructions, you can make that transition. And we have a discovery in the lab that we still don't understand, where we've done an experiment in cells where we've removed a particular important factor that models how the genome wraps itself around structures called nucleosomes. And we ask those cells to become heart cells, and they get halfway there, and then all of a sudden they take a left turn and they spontaneously become brain cells. It's absolutely nuts. And it goes completely against anything against everything that we think we know about how development proceeds. We always thought that once you acquire a certain potential in normal development, you're only going to make whatever should come out of that. So in the germ layer called mesoderm, you should only be making bone, heart, muscle, blood, for example. And so our cells, without that factor, they go and they make mesoderm, they go a little bit further, they look like they're going to become cardiac precursors, and then the next time we look at them, whoop, they're neurons. It's absolutely insane.
I find it's amazing. First of all, that there was a discovery so gigantic, just waiting to happen, right? And it's amazing, the work that you're doing. You never know when you're going to stumble on a breakthrough like that. And of course, there's a lot of hard work behind it. But I had a conversation with Shinya last summer when he talked about the importance of basic science. And he said, that is our strength at Gladstone. And he said half the textbooks are wrong. And we've got to fix that, not for our generation, but the generation after that. And so I think it's fascinating.
I mean, one of the things when you asked about the process of science and what you consider success, right? What I consider success, either from myself or from my colleagues, is disproving or providing an alternate interpretation to something that we thought we knew. Saying, well, we thought we knew this was like this, but it actually turns out it's completely different. And you kind of look at it and go, I guess you're right. So now, and that's great. That's really great, because as scientists, we're not dogmatic. We're not supposed to be dogmatic. We're supposed to be pragmatic. We are, and we accept that we don't know everything. And the discoveries like Shinya's, that you can revert back to a pluripotent state from terminal differentiation, was completely astounding. And it's created opportunities for disease treatments, but it's completely upended how we think about developmental biology.
Well, it's, for those of us who aren't scientists, it's just so inspiring to be able to spend time with you guys, and it's just rare to have this kind of mental flexibility and lack of dogma these days. So, Sarah, you want to come on and give us a sense of the questions that might have come in while we were hanging out?
Sarah, I work with Robert and Benoit. We got a ton of questions, so we should probably get right to it. The first one is super interesting. It comes from Steve. Thank you. Some of the descriptors that you used, such as sculpture, blueprints, parts, prompted him to wonder, given the advances in 3D printing, do you see a future where you can replace our entire hearts with a mechanical version?
Yeah, you know, that's something that a lot of tissue engineers are striving towards. And I think the more we learn about what you need to have, the more we should be able to do that. I think that the one of the things I think to sort of step back from is the notion that we need something that is shaped exactly like our heart. I mean, basically what we need something that pumps in, you know, with the right pressures to the lungs and to the body, and that can do so with, you know, automatically and continuously, and that is big enough to sustain the circulatory pressures that we have. So the challenges are that, you know, you need to, you know, the heart is complex in that it has a vast capillary network that sustains its oxygenation. And that's what I think is going to be the challenge, is to create a structure that not only is a big chunk of beating muscle, but is one that is properly vascularized. So I think we're I think far from it at the moment. I know there is at least one group that recently published sort of a very miniature version of what this could look like, but making it human size, I think, would be really, really hard. Mm-hmm. But it's not, I don't think that it's in the realms of the impossible.
Mm-hmm. Great. The next question is, do you think there will come a time when our understanding of the genetics of congenital heart disease allows us to correct heart formation through gene therapies such as CRISPR rather than surgery? And if so, should we do it?
Yeah, that's a really great question. I mean, the genetics serve many purposes, right? One is to understand the biology behind it. If we don't understand the biology, we don't know what kinds of treatments we could do. There are, in some cases, for example, the gene that we work on called TBX5, the kids that are born with those mutations have sometimes very severe limb defects, similar to thalidomide teratogenicity. And some of the parents who have Holt-Oram syndrome are deciding to do in vitro fertilization with pre-screening, right? So they screen for embryos to reimplant that won't have the mutation that they carry. But for that, you need to have absolute certainty about the causative nature of that mutation. What I'd like to see happening is to learn biology in a way from these mutations, and from then modeling the mutations in experimental systems, understanding the biology that they affect, to understand something akin to folate deficiency in neural tube defects, where you can say, okay, we've got these common pathways that maybe we can modulate, perhaps with this type of vitamin or diet or supplement or what have you, that we could prevent it from going the wrong way more often than not. Because the genetics of congenital heart disease are going to be very complex. Now, in terms of gene therapy with CRISPR, that is something that is not completely science fiction at the moment, but there are huge ethical issues around that we need to overcome.
Great. This next question is, my research is about understanding the development and differentiation of the endocardium that forms heart valves. What are your views on its specification in the early stages of mouse heart development? And what makes it different than the endothelium in terms of specification?
Wow, that's a
question I would have come up with.
Yeah. The endocardium is a really interesting one, because its origins very early on, I don't think are super well known. In our movies, we can see it. We can see the endocardium as it pops up, and it gets sort of wrapped into the heart tube. So I think it's initially contiguous with the vascular endothelium, but really acquires an identity of its own. And I think it's not quite understood at the moment what specifies it specifically in the early heart.
Great. Okay, there's a couple other ones. So what genes are you studying in the lab? And what techniques did you use to trace those cells?
So the lineage tracing that I showed was, so the in situ hybridization, the purple color, was a gene called TBX5, a transcription factor that's mutated in that syndrome with the limbs that I was talking about. And for the aficionados, we used a tamoxifen-inducible Cre recombinase to be able to genetically label those cells only at that time point, and then used a fluorescent reporter that turns on in the presence of active Cre to follow them later on. And actually, we got a bit fancier in that the red cells that are labeled in there are actually double labeled with another gene that uses a different recombinase. And so those report the double labeling of those progenitors.
Great.
And the other genes that we study are genes regulated to chromatin remodeling and histone modifications.
Sarah, I think maybe we'll take one more question because I'm mindful of Benoit's time and we want to get him back to thinking about science and what he's going to do when he gets him back into the lab. Do you want to pick one last question?
Yeah, this one is really interesting. As a PhD student that is about to graduate soon, they are very interested in your view on how to pick your postdocs. What features do you value the most? What makes your view about the current COVID crisis on current postdoc job market? Do you actively recruit postdocs now?
Yeah, well, we've had an issue with recruiting postdocs. We had one that was supposed to join May 1st and isn't coming. She was coming from Australia, so we'll have to wait on that. Looking for postdoc, I mean, you know, to some degree, it's the science that should drive how you pick a postdoc. But I think picking someone who is a generous mentor and who's created a culture of collegiality and collaboration within the lab, I think is probably the most important thing. You know, science is hard. And you know, it shouldn't be harder because, you know, somebody, you know, is a expects, you know, too much from people, or is a micromanager, or creates a culture of competition and so on. And I'm not saying that there's that much of it around, but there are some people who are really fantastic mentors, and you can, you know, it's easy to find them because people will talk about them as such. And you know, talking to the students in the labs, and you'll get a good feeling for what the PI is really like.
We just to wrap up, we had a few as we imagine this series over the next 3 sessions. We thought we might end by asking each of you the same 3 questions, like they did in the actor studio back in the day. And so just sort of speed round. What do you think science will be like in 10 years and in 50 years?
I think science in 10 years will largely be multimodal, real-time data collection and interpretation. I think we'll know everybody's genomes. We'll be able to have monitoring devices that will be like our usual, you know, well, now, you know, we talk about how the Apple Watch and those other devices can measure your heart rate and stuff. I think we're going to have devices that measure, you know, all your functions in real time. Will alert you if there's something not quite right. Will send messages to doctors for diagnosis. And then in terms of the science, I think we'll be able to, we'll have the tools to manipulate genes and proteins and organisms at will, far beyond what we can do now. And it'll be really a matter of understanding what all the new data mean. And so the folks doing computational biology are going to be more important than ever in 10 years.
If you could have the answer to one question, what would it be? Doesn't have to be science.
Oh gosh. That's a really tough one. Honestly, it is a science question. The one question I'd really like to understand is, how does a gene work, really? How does it work? Because we understand some molecular principles, but we don't really understand how a gene gets turned on or off and what it really means.
2 more minutes. What is something that you've seen over the last couple of months, or you've heard, or you've read that is going to stick with you? You are well known as a great cultural critic within Gladstone's ranks, and so give us your tip of the day.
Okay, there's the one that really struck me recently was Ravi Shankar's Morning Raga and Evening Raga record. That is just absolutely phenomenal. It is so peaceful, takes so much time, and the sitar goes and goes and goes and goes, and then at some point you're kind of lost in it, and then the tablas come crashing in, and it's better than a techno music rave drop. I mean, it's
This is work. You had the album and you just pulled it and started listening to it again.
Yeah, exactly. I just needed to clear my mind.
I am gonna dig it out.
That's that for me. And the other, I've been watching a lot of TV, and there's a lot of great shows out there. One that I find is particularly compelling because it is so on the edge of it not working, is Pamela Adlin's Better Things, which is it's just about family, about being together as family, and that really resonates now that I'm here with my family 24 hours a day, and she's a single mom with 3 daughters, and it's just so beautifully written, so heartfelt, and it's funny and it's sad, and she writes, directs, and acts in it, so it's all on her. And to me, it feels like how we're doing science, in that we're teetering on the edge of failure all the time. And when we make it happen, when it comes out at the end, and we're happy with the outcome, and other people are happy with the outcome, it's absolutely glorious. So her TV show, which is basically almost like a one woman show.
I'm going to watch it. I have not been watching TV. I hear that people have, but I don't know where they find the time. Thanks for taking us into your living room. This was more than I could have hoped for, and I bet that that's true for the people that tuned in.